Microbial production of human insulin for diabetes management. So what is insulin? Insulin is a peptide hormone produced naturally by the pancreas. Its main role is to regulate blood glucose levels. It does this by helping our cells absorb glucose, either to be used as energy right away or stored for later. Let's discuss the use of insulin in the human body. When we eat food like carbohydrates, they are broken down into glucose, which needs to be converted by insulin. The pancreas then releases insulin hormones to act like a key, where it signals cells in our muscle, fat, and liver to take up the glucose from the blood. Without enough insulin, glucose stays in the bloodstream and cells starve for energy. The body then breaks down fat and muscle for fuel. This leads to hyperglycemia, or high blood sugar. Over time, the lack of insulin or poor insulin function causes diabetes mellitus, also known as type 1 diabetes. For individuals with type 1 diabetes, the immune system destroys the pancreas' beta cells, so the body makes little or no insulin. Therefore, insulin therapy is essential for survival. Without insulin, blood sugar level rises dangerously, leading to critical conditions like dialysis, organ damage, or even death. That's why we use recombinant human insulin. It is a safe lab-produced version that mimics natural insulin. Now, let's look at how Malaysia handles this health crisis. We have Biocon Malaysia, which operates as a state-of-the-art integrated insulin manufacture and research facility. It is located in Johor. It is not only Malaysia's first and largest integrated insulin manufacturer, but also Asia's largest insulin production site. Biocon has already supplied over 100 million cartridge of recombinant human insulin to Malaysia's Ministry of Health, which has helped more than 345,000 patients. This achievement highlights how local production can improve access, lower costs, and strengthen the future of healthcare within the nation.
In fact, Biocon Malaysia has chosen Pichia pastoris with eukaryotic expression system as the host to produce recombinant human insulin. The main reason for this choice lies in the way yeast handles proteins. Insulin needs correct protein folding and disulfide bond formation to be biologically active. E. coli produces insulin. It often forms insoluble inclusion bodies that require complicated refolding steps. In contrast, Pichia pastoris secretes properly folded proteins, making the process simpler and more reliable. Another advantage is efficiency. Yeast is just like bacteria, grows rapidly but can achieve much higher cell densities. This enables large-scale fermentation with greater productivity and lower cost. Also, scalability is crucial. Pichia pastoris can be grown in large bioreactors under controlled conditions, ensuring a consistent global supply. At the same time, proteins expressed in yeast meet international quality standards, which help Biocon gain approvals in major markets, such as the US, Europe, Japan, and Australia. By choosing yeast, Biocon gained a strategic advantage. Unlike most competitors who rely on E. coli, Biocon's proprietary yeast platform offered a unique combination of cost efficiency and high-quality insulin production. Interestingly, all of this was made possible through recombinant DNA technology. The human insulin gene was inserted into Pichia pastoris using plasmid vectors. With the right promoters and regulatory elements, the yeast expressed recombinant human insulin, which was then purified and formulated for therapy. This resulted in a biosimilar insulin that is structurally and functionally identical to natural human insulin, ensuring both safety and efficiency for patient workflows.
Hi, I'm Bhuvanesh here. Today I'm going to talk about insulin fermentation technique, which is known as the methodology. In this technique, there are 5 main important steps. I'm going to talk about it, and also I'm going to share about the control parameters in this fermentation process. Insulin fermentation at Biocon Malaysia. Insulin, a globular protein essential for diabetes management, is produced at Biocon Malaysia using an advanced fermentation-based bioprocess. The facility employs a feed-batch fermentation system, a method commonly used in industrial recombinant protein production. Feed batch fermentation is ideal for insulin manufacturing as it allows nutrients to be supplied gradually, thereby supporting high cell densities, maintaining optimal protein expression, and reducing the accumulation of inhibitory byproducts such as acetate and ethanol. 5 main steps of insulin fermentation process by Biocon company. First one, gene insertion and host preparation. Second step, the seed train and fermentation. And third step, harvest. Fourth step will be downstream processing. Fifth step will be formulation and fill finish. Step 1, gene insertion and host preparation. The production of insulin at Biocon Malaysia begins with the genetic engineering stage, where the human insulin gene is inserted into a plasmid vector and then introduced into Pichia pastoris. This yeast serves as Biocon's proprietary fermentation host platform, chosen for its ability to express large amounts of recombinant protein efficiently and cost effectively. Next will be the seed train and fermentation process, which is known as the feed batch. Okay, small seed cultures are developed and gradually scaled up before being transferred into large bioreactors. A feed batch fermentation strategy is employed where nutrients such as carbon, nitrogen are supplied progressively rather than all at once. This approach allows the culture to maintain exponential growth and sustain high expression levels of insulin protein. Throughout fermentation, critical processes parameters including pH, temperature, agitation, speed, dissolved oxygen levels, and antiform control are closely monitored and regulated to optimize productivity and protein quality. The third process steps will be harvest. When the fermentation phase is complete and sufficient protein expression has been achieved, the culture broth containing the yeast cells and expressed insulin is harvested. This marks the transition from upstream to downstream processing. The downstream steps involve separation of cells, usually by centrifugation, followed by purification using chromatographic techniques such as ion exchange and reverse phase chromatography. The purified insulin is then crystallized to obtain its functional form and lyophilized to produce a stable powdered product that can be stored and processed further. The final stage is formulation and fill finish. Here, the insulin powder is reconstituted and formulated into injectable solutions. These are aseptically filled into multiple delivery formats, including vials, cartridges, pre-filled syringes, and insulin pens. Biocon's Malaysia's facility is globally certified for aseptic processing of biological, ensuring safety, consistency, and compliance with international regulatory standards. In summary, insulin production at Biocon's Malaysia follows a highly controlled and efficient pathway: gene insertion, feed batch fermentation, harvest, downstream purification, crystallization, lyophilization, and final sterile formulation. Key parameters such as pH, temperature, dissolved oxygen, and sterility are strictly regulated at every stage, ensuring high yield and quality. Through this approach, Biocon Malaysia has become a global leader in affordable insulin manufacturing, contributing significantly to improve access and affordability of this life-saving drug worldwide. Control parameters during fermentation process: pH between 5 to 6, temperatures between 28 to 30 degrees Celsius, dissolved oxygen above 30%, agitation between 300 to 900 rpm, and aeration at 0.5 to 1.5 ppm. And then anti-form automated dosing to control form formation, aseptic environment, HEPA filtered air, purified water system, environmental monitoring, building management system.
Insulin is one of the most essential biopharmaceuticals in the world. Now let's look into the target market segments.
The
primary market segment is patients with diabetes. According to the International Diabetes Federation, over 530 million adults worldwide are living with diabetes, and this number is projected to rise to 783 million by 2045. The largest need is in low and middle income countries where affordability is a critical issue. At the same time, developed markets like the United States and Europe are increasingly adopting biosimilar insulins to reduce healthcare costs. Biocon Malaysia plays a central role in this market. The Johor facility exports insulin to over 120 countries with a focus on Asia, Africa, and Latin America. These regions represent underserved markets where high costs have historically limited access. By offering biosimilar insulin at much lower prices, Biocon is helping to close the treatment gap. Demand for insulin is rising steadily. The World Health Organization estimates that insulin demand will grow by more than 20% in the next decade, driven by the global rise in diabetes cases. Biosimilars are especially attractive because they are clinically equivalent but more affordable than branded insulin. Biocon's recent $250 million expansion in Malaysia, which will double production capacity, reflects confidence in this growing demand. However, there are several challenges. From a technical standpoint, producing recombinant insulin at large scale requires high precision in fermentation and downstream processing. Ensuring batch-to-batch consistency, product stability, and compliance with good manufacturing practices is a continuous challenge. Economically, while Biocon aims to make insulin affordable, the cost of research, clinical trials, and large-scale production remains high. In many developing countries, healthcare budgets are limited, creating strong pricing pressure. Biocon also faces competition from global insulin leaders such as Novo Nordisk, Eli Lilly, and Sanofi. On the regulatory side, approval for biosimilar insulin requires extensive data to demonstrate safety and equivalence. Regulatory processes vary across regions, which can delay market entry. Clinical studies and compliance with multiple agencies such as the FDA, EMA, and WHO add both time and cost to commercialization. To overcome these issues, Biocon continues to invest in process optimization and automation to lower production costs and partners with governments and NGOs to expand affordable access. Harmonizing regulatory frameworks will also speed up global distribution. In summary, the insulin market is growing rapidly, but companies must navigate technical, economic, and regulatory hurdles to succeed.
If someone asks why Malaysia is key for insulin production, I will say that Malaysia offers a unique combination of advantages, like a strong government push for biotechnology, a skilled workforce, and world-class infrastructure that supports large scale of biomanufacturing. At the same time, Malaysia is located in the heart of Southeast Asia, where diabetes is a growing public health challenge. By producing insulin locally, Biocon not only supplies Malaysia, but also becomes a strategic hub to serve millions and millions of patients across Asia and beyond. In short, Malaysia is not just a place to manufacture, it's the right place to make insulin affordable, accessible, and sustainable for the future.
To conclude everything from this video, the microbes help to diagnose, isolate, and prevent harmful bacteria from spreading or even infecting new people. It can also be used in medicine to combat against illnesses. Hopefully, in a possible future, microbes can help with being used in a variety of methods to keep people healthy, like combat against cancerous cells before any serious cancer happens, and requiring learning on how these microbes work in certain environments of the human body, maybe in changes of temperature or the pH changes. Microbes are a definite benefit for healthcare to those who are in desperate need of it, along with helping with living a longer and better life from them.
1, 2, 3 mic test. Fact, Biocon Malaysia has chosen Pichia pastoris.
Today I'm going to talk about fermentation. Wait.
Insulin is one of the most